let's come to the last part of this particular sequence analysis. And it's been several modules for this. Clearly, we have to understand that in terms of sequence analysis, uh, there is a lot more, and the next set of modules after this will go into genomics, which is, you take the scaled down version of a sequence. And if you remember, this started with a module on explaining what a DNA structure is. So when you write the primary sequence, the sequence of letters A, T, C, G, etc., understand what kind of structure is behind it. So cladistics is a particular method of hypothesizing relationships among organisms. Like other methods, it has its own set of assumptions, procedures, and limitations. Cladistics is now accepted as the best method available for phylogenetic analysis. There's that word again, for it provides an explicit and testable hypothesis of organismal relationships. So again, just reading this, the basic idea behind cladistics is that the members of a group share a common evolutionary history and are closely related to members of the same group when compared to other organisms. So you look at, for example, when you think in terms of cladistics, you think about, oh, look at them, they share a unique feature, so we can clump those organisms with similar features together, and these shared characteristics. But remember the word, not just shared, but derived characteristics are called synapomorphies. Okay, so again, if we say, well, just because you have a dark-haired human and a chimpanzee, for example, has dark hair, we don't just say, oh, this is a synapomorphy, so they must be related in some way. They are related, we understand that, but that relation is more evolutionary, as opposed to saying, well, we're just going to clump everything because they have black hair, for example. Okay, so that's why the characteristics are shared, but the characteristics are also derived. And where are they derived from? They derive from a common ancestor. Okay, so assumptions of cladistics: any group of organisms are related by descent. So remember, there's a common ancestor, which goes back to the kind of a silly example I gave between humans and, for example, a chimpanzee just being clustered together, even if there's no evolutionary relationship, just because they happen to have black hair or dark hair. Okay, so the first assumption is made for all evolutionary biology. So literally, you would say all of every species from the beginning of time is part of a clade. Okay, that means that life arose on Earth only once, and therefore all organisms are related in some way by descent, because everything shares a common ancestor, no matter how primordial that ancestor was, no matter how primitive that, whether everything arose from a unicellular organism. All of creation, in terms of the animalia, can come from a single species. Okay, and so all of it can fall into a single clade. There is a bifurcating pattern of cladogenesis, that is, new kinds of organisms may arise when existing species or populations divide into exactly two groups. So it's bifurcating. Okay, and so what happens is, now you might have a parent, and the parent splits into two daughters, and then these daughters split further into two daughters, and along the way there might be a relationship where the split occurs a little earlier, but it's always a bifurcating split. Okay, always divisions by two. And changes in characteristics occur in lineages over time, that the characteristics change over time, and this is really important. And it is only when these characteristics change can we say, oh, we see a split. Okay, we see a split. And let's look at some examples. So the point is, once you understand that there is a split, let's think about cladograms, which is another way of organizing things by evolutionary descent, just like phylogenetic analysis. But there are some differences. So cladistics is useful for creating systems of classification. Cladistics predicts the properties of organisms, and this comes from this notion of synapomorphies. Cladistics helps to elucidate mechanisms of evolution. Okay, so now we talk about cladistics. What is a clade? A group of organisms that arose from a common ancestor. Okay, remember when we talk about the primordial species or primitive species, and if that was that unicellular organisms was the beginning of everything, based on whether you think in terms of new species arose because of gradualism, which is there's always a big debate, gradualism, where where species arose gradually, whereas versus punctuated equilibrium, where there was these not a lot of new species being formed, and then there's something something epigenetic, something environmental that took place, and that resulted in a lot of species, and then everything kind of stayed the same, and then again there was another huge something else happened that caused a whole bunch of mutations and then caused a whole bunch of speciation. Okay, so a group of organisms that arose from a common ancestor. So there's this different things of what are called taxons. Okay, so a monophyletic taxon is a taxon where you have a bifurcation that results from a common ancestor is a bifurcation. And similarly you can see this entire clade can actually arose from a common ancestor. The next example is of uh, a polyphyletic taxon, where you see there is a clade, that is synapomorphies or similarities arose from species that come from two different organisms. Okay, that is polyphyletic. And paraphyletic is where the bifurcations, etc., can all be traced to a common ancestor. So, for example, this clade consisting of J and K, yes, they arise from a bifurcation, but they can be traced to a common ancestor. So that's paraphyletic. Okay, so monophyletic, polyphyletic, and paraphyletic. Here's a primate cladogram. And so, for example, remember, it's always bifurcations. And you can see the lower order versus the species as they evolve. So if we had a common ancestor, you realize that we go to these to humans, but along the way, you see the bifurcations that occur. And so we have closest are the bonobos and chimpanzees. A little back in time is gorillas, orangutans, gibbons, and siamangs. And you'll see something that's a lot more primitive, but the good part about the clade is because we're looking at synapomorphy, so we're not really concerned with kind of evolutionary timescales. We're just concerned with the end result. So the point is, though a split occurred which resulted in lemurs and lorises a lot earlier, so you would say, well, there's that kind of these things stayed primitive, whereas this branch ended up as a lot more advanced, an advanced species like humans and apes. You understand that we are really concerned with the end result here. Okay, now you can also think of this as a primate phylogenetic tree. Now, a phylogenetic tree is important because we are not just concerned with the end result, but we're also concerned with the timescales. So, what you have here is humans, chimpanzees. The While though the bifurcations occurred earlier, you will see that um, you will see that all these species actually exist. Okay, they actually exist in kind of in their modern forms. Now, the good part about phylogenetics is that you can actually have, for example, an x-axis here, as I'm tracing with a mouse, an x-axis here, and that x-axis can be time. Okay, and then you can actually scroll this way, you can kind of draw these lines where you can actually see at what point in time, based on this evolutionary parent, at what point in time these bifurcations occurred. And then you can actually say, well, here is at this point in time is where this species changed, and one of it resulted in the apes, and the other one resulted in the old world monkeys. Okay, the new world monkeys, and then you have lemurs, which are essentially prosimian. Okay, on the other hand, you have to remember that lemurs do exist. Okay, they have, just because they were prosimian, as species kind of changed and speciation occurred, the lemurs are as primitive, but they still actually exist, and that's really important to understand. So a phylogeny or an evolutionary tree represents evolutionary relationships among a set of organisms or group of organisms called taxa. The tips of the tree represent groups of descendant taxa, often species, and the nodes represent the common ancestors, right? So, for example, if you go back, here's a node. Here's a node where a baboon and a mangabey split, right? So this is that tells you the evolutionary consequences, and these points where the bifurcation can is occurring are actually the nodes. Um, Two descendants that split from same node are called sister groups. In each tree below, species A and B are sister groups. They are each other's close relatives. So what's the difference between a phylogenetic trees and cladograms? Very often the terms are used interchangeably and incorrectly, and we've seen that quite often, right? Our first discussion was on bioinformatics, bioinformaticians, bioinformaticists. Then there was a big discussion on the differences between bioinformatics and computational biology. Okay, so these are interchangeably. A tree and its branches represent parent, daughter, sister, sister relationships representing ancestry. With cladograms, the length of the branches do not mean much in terms of evolution. They're just connections. But in phylogenies, the length of the branches do mean evolutionary timescales. When a taxonomy, for example, where did the speciation occur? Where did the bifurcation occur? At what point in time, what point of time does a node exist which has evolutionary consequences because a parent organism speciated into two daughter organisms. Uh, and here's a very interesting, it's kind of an old world style thing, which actually gives you a sense in terms of the time scales are on the y-axis, and you see that if you start from a primitive organism, you can actually say, looks like the branches of a tree, but this is where this is basically a clade, and here are where evolutionary. These are some of the. This is how these things evolved. Now. Um, so you can think in terms of the Cambrian, Silurian, Devonshire, Permian. Then you have the Triassic, and so you might say, well, there were fish which existed, and then it almost like a lot of fish, a lot of speciation, and then the fish kind of stabilized into the fish that we see today. A lot of bifurcation, and then you see a lot of where these branches went nowhere. That's because the fish, for whatever evolutionary reason, they died. They died off. They became extinct, and then you have a lot of frogs, and then a lot of these died. You see the tree branches going nowhere, and then the ones that we know as frogs and toad families have persisted till this day. But you can see in terms of the Paleozoic era is where amphibians were really a lot, and you can also see how, for example, one species is persists, and then so as you see, as the fish kind of go kind of become less fewer and fewer, with a lot of extinctions taking place, the reptiles increase. And this time the rhinocephala, that is the kind of thing species with the brain, they were merely a single branch, and they didn't even arise, right? So marsupials, placentals, and then you think of the Cretaceous, the Jurassic, and the Triassic period, where the reptiles were dominant, right? And when you think in terms of reptiles, we think in terms of the dinosaurs, right? So this is the Jurassic. Remember Jurassic Park. So the Triassic dinosaurs were primitive dinosaurs, and really the Jurassic area is where the reptiles were a lot, and then a lot of these branches went nowhere. The animals, many species became extinct, and now it's somehow kind of rationalized and stabilized into the few reptiles that we see. But you see, the Mesozoic period was such an incredible age for reptiles. And then the reptiles split into birds, and again, the birds, have remained relatively consistent. There's not been a great kind of increase and decrease. And then you see how, for example, as the fishes stabilized into the ones that evolutionary survived, the amphibians survived a lot fewer than what they started out with during the Paleozoic era. And now what has happened is the mammals have, at this point in time, the Cenozoic era, the mammals have proliferated. Obviously, man, it'll be interesting to see exactly where this goes on, right? So this is very interesting. The Archaeozoic era, a lot of the Archaea, a lot of very protozoans, very unicellular, and then you see how the branching takes place. So this is a terrific resource, which gives us a sense of how some of this arose. And again, like we talked about, the going back to the primates, you actually can along the x-axis put millions of years on the x-axis, and the phylogenetic tree, as opposed to a cladogram, will tell you. You can actually do a count and say exactly where do humans arise. Um, so for example, humans. So you had a bifurcation here between what gave rise to humans and some of the other higher apes, and then 0.84 million years later, the humans arose. On the other hand, you had kind of another bifurcation which gave the pygmy chimpanzee versus the common chimpanzee. So you see, you can go backwards in time and you see where that bifurcation arose. Uh, this is all we have in terms of sequence analysis. Thank you.